Welcome to all students. Myself, Om Prakash Godara, Assistant Professor for Physiology Department, Faculty of Agriculture and Veterinary Science. Today, we discuss about our topic crop growth analysis. Mainly, growth analysis can be used to account for growth in terms that have functional or structural significance. The type of growth analysis require measurement of plant biomass and assimilatory area, leaf area, and method of compounding certain parameters that describes growth. The growth parameters that are commonly used in agriculture research and the name of the scientist who proposed the parameter are given below. Leaf area index scientist will mainly given below Williams 1946 and leaf area ratio Redford's 1967 and leaf area durations Power at 1967 SLA Kawit et al 1971 and SLW Pierce et al 1968. Net assimilation rate Williams 1946 and crop growth rate Watson 1956. Relative growth rate Williams 1946 and harvest index Nikitrovich 1951. Mainly leaf area. Leaf area क्या होता है? तो leaf area this is the area of photosynthetic surface produced by the individual plant over a periods of intervals of time and expressed in centimeters square per plants. Leaf area index Williams 1946 proposed the term leaf area index. It is the ratio of the leaf of the crop to the ground area. जो मेनली रेशो होता है लीफ एरिया का वो ग्राउंड लेवल की तुलना में कितना है और पीरियड ऑफ इंटरवल ऑफ टाइम के अकॉर्डिंग इंटरवल के अकॉर्डिंग जो रेशो होता है वो मेनली जो लीफ एरिया है द वैल्यू ऑफ लीफ एरिया इंडेक्स शुड बी ऑप्टिमम एट द मैक्सिमम ग्राउंड कवर एरिया at which crop canopy receive maximum solar radiations, hence the TDMA will high. लीफ एरिया इंडेक्स मेजर्स टोटल लीफ एरिया ऑफ प्लांट का जो टोटल लीफ एरिया होता है और अपॉन ग्राउंड एरिया ऑक्यूपाइड बाय द प्लांट्स के द्वारा जो ग्राउंड का कितना एरिया वो ऑक्यूपाइड किया गया leaf area ratio the term leaf area ratio was suggested by Redford's 1967 express the ratio between the area of leaf lamina जो लीफ लेमिना होती है उसके द्वारा जो एरिया कवर किया जाता है और टोटल प्लांट बायोमास or leaf area ratio reflects the leafiness of a plant or amount of leaf area formed per unit of biomass and expressed in centimeter per square per grams of plant dry weight. Leaf area ratio measured leaf area per plants upon plant dry weights. Leaf weight ratio. It was coined by Kavit et al.'s 1971. Leaf weight ratio is expressed as the dry weight of leaves to wool plant. Dry weight and is expressed in gram per grams. Mainly leaf weight ratio, leaf dry weight upon plant dry weight. After that, leaf area durations to correlate dry material with leaf area index. Power et al.'s 1967 integrated the leaf area index with time and called as leaf area durations. Leaf area duration take into accounts both the durations and extent of photosynthetic tissue of the crop canopy. The leaf area durations is expressed in days. Mainly, it measures leaf area durations L1 plus L2 upon 2 multiplied by T2 minus T1. L1 is equal to leaf area index at the first stage and L2 is the leaf area index at the second stage and T2 minus T1 is equal to time interval in days. Specific leaf area SLA. Specific leaf area is measure of the leaf area of the plant of leaf dry weight and expressed in centimeter square per grams as proposed by Kavit et al.'s 1971. Specific leaf area measures leaf area upon leaf weights. Hence, if the specific leaf area is high, the photosynthesized surface will be high. However, no relationship with yield could be expected. Specific leaf weight SLW. It is a measure of leaf weight per unit leaf area Hence, it is a ratio expressed as gram per centimeter square, and the term was suggested by Pierce et al. 1968. More specific leaf weight upon unit leaf area indicates more biomass and positive relationship with yield can be expected. It is measure specific leaf weights upon leaf area. Absolute growth rate AGR. AGR is the functions of amount of growing material present and is influenced by the environments. It gives absolute value of biomass between two intervals. It is mainly used for a single plants or single plant organs. Example, leaf growth and plant weights. It's mainly absolute growth rate measures S2 minus H1 upon T2 minus T1 centimeter per day, where H1 and H2 are plant heights and T1 and T2 times respectively. Net assimilation rate, the term net assimilation rate used by Williams 1964. Net assimilation rate is defined as dry matter increments per unit leaf area or per unit leaf dry weight per unit of times. The net assimilation rate is a measure of average photosynthetic efficiency of leaves in a crop community. It is measures net exponentials W2 minus W1 upon T2 minus T1 multiplies log L2 minus log L1 upon L2 minus L1, where W1 and W2 is dry weight of whole plants at a time T1 and T2 respectively. L1 and L2 are leaf weight or leaf area at T1 and T2 respectively. T1 minus T2 are time interval in days. Net assimilation rate is expressed as the grams of dry weight increase per unit dry weight or area per unit times grams per day. Relative growth rate, the term was coined by Williams 1946. Relative growth rate expresses the total plant dry weight increase in a times interval in relation to the initial weight or dry matter increment per unit biomass per unit times or grams of dry weight increase per gram of dry weight and expressed as unit dry weight per unit dry weight per unit times. It is measures relative growth rate log W2 minus log W1 upon T2 minus T1, where W1 and W2 are whole plant dry weight at T1 and T2 respectively. T1 and T2 are time intervals in days. Crop growth rate. The method was suggested by Watson's 1956. The crop growth rate explains the dry matter accumulated per unit land area per unit times gram per meter square per day. Crop growth rate measures W2 minus W1 upon del T2 minus T1 when W1 and W2 are whole plant dry weight at time intervals T2 T1 minus T2 respectively. So is the ground area on which W1 and W2 are recorded. CGR of a species are usually closely related to interceptions of solar radiations. Today, total dry matter productions TDMP and its distributions. The TDMP is the biomass accumulated by the whole plants over a periods of intervals of time and its distributions via allocations to different part of the plant, such as root, stem, leaves, and the economic part which control the sink potential. Translocation percentage. The term translocation percentage indicate the quantum of photosynthesis translocated from source का मीन्स होता है extra to the grains का मतलब होता है sink जिसको अपन पेनिकल्स या grains बोलते हैं from flowering to harvest. Mainly translocation percentage is measures extra weight at flowering minus extra weight at harvest upon penicle weight at flowering minus penicle weight at harvest. इनका जो डिफरेंस होता है फ्लोरिंग स्टेज पे और हार्वेस्टिंग स्टेज पे एक्स्ट्रा और पेनिकल्स का उनका डिफरेंस जो मेनली आपका ट्रांसलोकेशन लाइट extinction coefficients. It is the ratio of light intercepted by crop between the top and bottom of crop canopy of to the leaf area index. Mainly K is equal to log I upon I0 upon leaf area index, where I0 and I are the light intensity at top and bottom of a population with leaf area. leaf area index. Leaf transpiration ratio LTR. It is expressed as the ratio of quantum of light intercepted by crop canopy at the top to the bottoms. Light intensity is expressed in K lux or watt per meter square. So LTR is equal to I upon I0, where I light intercepted at the bottoms of the crop canopy and I0 light intercepted at the top of the crop canopy. Dry matter efficiency, it is defined as the percent of dry matter accumulated in the grains from the total dry matter produced over the crop growth periods. Dry matter efficiency measure grain yield upon TDMP multiply so upon durations of crops. Unit area efficiency. Unit area efficiency, it is expressed as the quantum of grain yield produced over a unit land area for specified crop growth periods. U- Unit area efficiency measures grain yield upon land area multiplied 1 upon durations of crop. Harvest index, the harvest index is expressed as the percent ratio between the economic yield and total biological yield and was suggested by Nikiforovich 1951. Harvest index measures economic yield upon total biological yield multiplied by 100. सो हार्वेस्ट इंडेक्स से मेनली इसमें जो आपका टोटल बायोलॉजिकल यील्ड है इसके अंदर मेनली आपका इकोनॉमिक यील्ड प्लस जो एक्स्ट्रा है उसको इन्वॉल्व किया जाता है और इस तरीके से अपन कोई भी जो क्रॉप है उसका जो हार्वेस्ट इंडेक्स है वो मेजर कर सकते हैं। तो इस प्रकार मेनली जो प्लांट है उसकी जो ग्रोथ एनालिसिस है वो डिफरेंट से जो फैक्टर है उसके द्वारा अपन मेजर कर सकते हैं। तो इसका एक इम्पोर्टेंस रोल रखता है कि प्लांट है उसकी जो यील्ड है उसका जो परसेंटेज है या जो उसकी फोटोसिंथेटिक रेट है वो मैंने लीफ पे किस प्रकार मेजर करती है या उसका जो लीफ एरिया है लीफ एरिया इंडेक्स है रिलेटिव ग्रोथ रेट है क्रॉप ग्रोथ रेट है नेट एस्टिमेशन रेट है ये डिफरेंट जो रेट्स है उसको अपन किस तरीके से मेजर कर सकते हैं। तो इसका मेनली डिफरेंट जो फॉर्मूलास है उसके द्वारा मेनली इसको अपन आइडेंटिफाई किया जा सकता है। तो मेनली जो ग्रोथ एनालिसिस है ग्रोथ का वो मेनली लेबर से होता है। तो लेबर के अंदर ये मेनली आप इजी वे चीज को निकाल सकते हो। तो मेनली जो क्रॉप ग्रोथ एनालिसिस है इसके लिए मेनली आपको कुछ जो फॉर्मूलेज हैं उसका आपको यूज करना पड़ता है। तो कि आपको लीवर इंडेक्स निकालना है क्रॉप ग्रोथ रेट या आपका नेट एस्टिमेशन रेट किस तरीके से मेजर करना है तो उसके अकॉर्डिंग मेनली अपन जो प्लांट है उसकी जो ग्रोथ है उसका एनालिसिस कर सकते हैं। ओके थैंक यू स्टूडेंट्स।